Welcome to a new episode of Drops of Science, the multilingual podcast that brings you the latest news and discoveries in the world of natural sciences. We begin this episode by discussing how octopuses move, particularly their arms, which are not to be confused with tentacles, typical of squids. Their arms, along with their suckers, make up a large portion of these animals' body mass. They also contain extraordinary neural and muscular adaptations. The peripheral nervous system holds most of the animal neurons and is largely concentrated in the arms. Moreover, the hydrostatic muscular structure, composed of 4 muscle groups intertwined with connective tissue, grants these animals unmatched flexibility. But do all the arms work the same way? To find out, a team of international researchers filmed 25 wild octopuses in Spain and the Caribbean between 2007 and 2015. From these videos, they extracted key information on arm movements. The scientists analyzed 3 levels: the overall behavior of the animal, the specific arm actions, and the deformations that allowed each movement. All of these was summarized in a detailed ethogram. 12 actions were identified, such as lifting, extending, and grasping, consisting of 4 deformations: shortening, elongation, bending, and twisting. In total, almost 4,000 actions were recorded, and all 8 arms were able to perform them. Not only that, a single arm can perform multiple movements simultaneously, and different arms can act at the same time. When analyzed in pairs, no dominance emerged between right and left. Octopuses are ambidextrous, working with coordinated pairs. However, Dr. Richter and collaborators observed a division of tasks. The front arm were used more often, about 64% of the time, especially for exploration and reaching, while the back arms were better suited to locomotion, such as lifting the body or rolling across the seafloor. Understanding how small local deformations generate such complex movements not only helps us read octopus behavior better, but also inspires soft robotics. This discipline draws directly from their flexible arms to design tools capable of moving and grasping delicately in challenging environments, from rescue operations to medicine. The next steps are to compare species with different morphologies, such as those with extremely slender arms, to see how shape affects the division of labor, and to understand how this knowledge may lead to new bio-inspired technologies. We now move to the surface to discuss an incredible adaptation some plants have developed to cope with excessive heat. In nature, there's a phenomenon called floral dimorphism, whereby some plants change the color of their flowers seasonally. Moricandia arvensis is a perfect example. It bears lilac spring flowers and white summer flowers. Previous experimental studies show this color change is highly plastic and mainly triggered by summer-like temperatures between 30 and 35 centigrades, which activate the production of white flowers. Across seasons, the pollinators visiting the plant also seems to change. A research team therefore investigated the intrinsic reasons behind these temperature-driven color shifts and whether they affect how insects perceive the flowers. To explore these, in 2019, researchers collected 1 lilac spring flower and 1 white summer flower from 100 plants of Moricandia arvensis at Negratin in Spain. Petals were collected and, while still fresh, processed in a mobile lab to measure reflectance spectra, meaning quantify colors, and extract pigments. From the spectra, they reconstructed how pollinators such as bees, flies, butterflies, and beetles see petal color, and estimated how much the flower stands out against spring background, grass, and summer background, soil, using chromatic and achromatic contrast. In parallel, pigments were quantified and identified. Results obtained by Dr. Narbona and collaborators showed that insects do perceive different colors, and background matters. In summer, the flower stands out more for bees and flies, whereas in spring it's more evident for butterflies and beetles. The color shift arises because in spring, anthocyanins dominate. They absorb visible light and are the reason we see those petals lilac. But in summer, they almost vanish, while UV-absorbing flavonols increase, and UV light is invisible to us, but visible to many insects. Anthocyanins are thermolabile. The heat slows their synthesis and increases degradation, whereas flavonols rise, acting as a sunscreen and limiting radiation damage. The color change is likely a plastic response to rising temperatures and UV radiation, a physiological defense that triggers an ecological response, since different pollinators recognize and visit the plant more at different seasons. The next 3 stories will focus on newly discovered species, whose identification, however, occurred for very different reasons. Let's start by talking about giraffes, which at first glance might all seem the same, but today we know that there are more species of giraffe than we might think. A recent review by the International Union for Conservation of Nature, based on original and updated data as well as expert opinions, has recognized the existence of 4 distinct species and several subspecies. The number of giraffe species has always been a matter of debate among scientists worldwide. In fact, in 2016, it was believed that there was only 1 species with as many as 9 subspecies. So, what led to this update? An improvement of genetic and genomic analysis carried out on existing giraffe populations, made possible by recent scientific advances. The assessment was conducted using the so-called traffic light system, already applied to other animal groups, such as canids and felids. This method combines multiple converging lines of evidence: morphological phenotypic characteristics, meaning physical traits distinguishing one population from another, genetic data to reconstruct close and distant relationships, and biogeographical information, showing how populations have been distributed and separated in the past and the present. The traffic light categories are distinct, green, where 3 or more independent lines of evidence indicate clear differences; probably distinct, yellow, where multiple lines of evidence suggest differences, but further research is needed; unknown, invalid, gray, little or no available evidence; not distinct, red, where there is no evidence of separation or published studies confirm conspecificity or subspecificity. A key contribution to this review came from 3D morphometric analysis of 515 giraffe skulls from across Africa. By contrast, the coat pattern, although distinctive, is not considered a reliable criterion, as its shape and color vary with the age and population. The combined data clearly distinguished, green light, the following species, with their respective subspecies: Giraffa camelopardalis, with 3 subspecies; Giraffa reticulata; Giraffa tippelskirki, with 2 subspecies; and Giraffa giraffa, with 2 subspecies. These taxonomic updates are crucial for conservation, as they provide a clearer picture of giraffe diversity and offer a solid foundation for targeted management of their populations. The next species we will talk about were found in the deep ocean. We still know very little about the living beings that inhabit the ocean waters surrounding our planet. It is estimated that more than 80% of the oceans have not yet been mapped or explored. Therefore, every new discovery adds a building block to our limited ocean knowledge. In this context, thanks to a 2019 expedition led by Dr. Geringer, 3 new species of snailfish were identified. Snailfish, from the family Liparidae, live in temperate to cold habitats across all ocean basins, playing very important roles in ecosystems. Their common name comes from the fact that most of them have a specialized ventral suction disc used to cling to rocks. When this happens, the animal adopts a curled posture, much like land snails. These animals are found both in shallow waters and in trenches exceeding 2,000 meters in depth, areas that are difficult to explore. The specimens collected in 2019 lived at depths between 3,268 and 4,119 meters in the Eastern Pacific, and their identification was made possible through tissue collection, DNA amplification and sequencing, microscopy, and microcomputed tomography. Evolutionary analysis made it possible to compare their genetic data with those of the other species, and to confirm that they are indeed 3 new species. The first is Careproctus colliculi. Pink in life, it has 22 pectoral fin rays, a rounded head, 8 caudal rays, large eyes, and well-developed pterygophores, bones or cartilages that articulate the base of the median fin rays, forming a large suction disc. Its scientific name refers to the Greek name of the small characteristic bumps that cover the skin, especially the head and the front of the fish. The second species, Careproctus yanchei, has a moderate ventral suction disc, single nostrils, and 6 brachiostegal rays. It has an entirely black body, rounded head, and horizontal mouth. The species is dedicated to Dr. Paul Yancey of the Whitman College in the United States for his contribution to deep-sea biology. The third species is Paraliparis M, with a long, black, laterally compressed body. It lacks a suction disc, has a singular pectoral fin ray, an anus anteriorly positioned, and a strongly angled jaw. The species is named after the collection site, Station M, paying tribute to all the researchers who have worked there. These discoveries help to fill gaps in abyssal sampling and remind us of the importance of integrating observations, morphology, and genetics to understand marine biodiversity. For the last species, we move to Texas, among the birds, where it appears that climate change has encouraged the emergence of a hybrid between two well-known bird species in that region: the green jay, Cyanocorax incas, and the blue jay, Cyanochyta cristata. The green jay is a tropical species, whose known distribution extends from the northern Andes through Central America to North America, where it is found in Mexico and southern Texas. The species has expanded its range significantly over the past 20 years, shifting farther in latitude, likely due to the rising temperatures in Texas and the absence of prolonged periods of cold winters. The blue jay, on the other hand, has moved its range. While it was previously found in the eastern United States as far south as Houston, it is now only found in South and West Texas, thus creating a range that overlaps with that of the green jay. The two subspecies belong to two completely different genera. Their common ancestor, in fact, must have lived around 7 million years ago. The discovery of the hybrid occurred almost by chance, when Dr. Brian Stokes was monitoring web pages dedicated to bird sightings in the United States. In particular, on the text bird page, at the end of May, he noticed that someone had posted an image of a bird with mixed characteristics. Likely, he was able to meet the photographer, set up fine mesh nets in his garden, and capture the bird. Before the capture, the researcher observed that this bird had closely followed a flock of blue jays for 2 days, and that it could produce vocalizations similar to the species, usually in response to the blue jay's vocalizations and activity. The hybrid also produced big clicking calls and low two-tone rattles, typical of green jays in Texas. Once captured, blood was extracted, the animal was ringed and then released. Genetic DNA analysis confirmed the hybridization. Specifically, it was determined to be a male hybrid, the offspring of a green jay mother and a blue jay father. The discovery may be an isolated event, as no other specimens have been cited to date, but it adds to a list of species interactions that are emerging as a result of climate change. It is urgent to document these new interactions, as they could lead to major changes in biotic communities. This news concludes today's episode of Drops of Science. Thanks for staying with us. See you soon, and good science!